morning. Today I want to talk about my PhD project. My name is María Fernanda Mideros. I am a PhD student of biological science. And in this moment I am working with evolution and population genetics of plant pathogens. This presentation will be used during my quality test in 2 months. And I want to talk about some aspects of the evolutionary relationship and adaptation to solanum potassium of newly emerging Phytophthora sensilato plant pathogen. So to start, I want to talk about Phytophthora. This is a microorganism that you can see here in this picture, some symptoms of this plant pathogen. This is really important because this is the agent for the one most important disease in potatoes and tomatoes. Phytophthora is the most destructive in crops, and this is an estimation about the losses that cause this plant pathogen. In this picture, for example, you can see here the symptoms of this plant pathogen. This is a potato and tomato, and after this pathogen arrives to a crop, cause these symptoms. And it's really important to study the evolution of this pathogen because, for example, in animal and human pathogens, the invasions of new disease has been very sad. But in plant pathogens, the studies of this is less common. This is the history of this pathogen. This is the agent of Iris potato famine. Iris potato famine was the most important emerging disease in the 19th century. As you can see here some pictures, because the people only eat potatoes, and this pathogen was the most devastating microorganisms. In this map, you can see the emergency of new disease. For example, this is very common in Europe, in North America, and in South America is very common in the tropical regions. In South America, this emerging plant pathogen causing negative economic impacts, and it's very interesting because in South America this pathogen could produce the disease in new hosts. In this case, for example, in tree tomato or solanum potassium, in Naranjilla lulo, in solanum quitoense, or for example in Chua is very common, this kind of pathogen. So recently, different authors have been proposed that this pathogen is a new species. As you can see here, the last publication of this pathogen, are the symptoms and the morphological feature of these microorganisms. In Colombia, you can see here the effect of this pathogen. This is a crop of solanum potassium, and you can see the symptoms of these microorganisms. So we consider that the epidemiological and evolutionary process underlying the introduction of this pathogen is really important because we need to know the history about the introduction of this microorganism in Colombia. So in this moment I am working with a collection of several isolates. This collection were doing in Nariño and Putumayo. We have 100 isolates. This is only pictures about the locations of these samples. For this analysis, we use microsatellites genetic markers. This is a small region in the DNA that is able to identify several selection results. This is the result of this analysis, and you can see here this is a distribution of isolates. In red in red box, you can see the isolates from tree tomato. In this case, these isolates are very different. You can see here that the red box is separate from the other isolates. And this is the analysis of the same markers. As you can see here, that these populations are separate from another population of this pathogen. And the results may conclude that this population is a new pathogen in Colombia. In conclusion, this is a preliminary analysis. Don't see a clear relationship, but the results show that this is an emerging pathogen. In addition, we use several aggressive tests. In this case, we inoculate the leaf of tree tomato. As you can see here, this is development. This is the day 6, 7, 8, and 9, and you can see here, for example, that the pathogen causes necrosis in the left. So, results of cross-host pathogenicity test show that all strains of these pathogens, it's very specific and it's very aggressive. This is the same in field. You can see, for example, in 2 or 3 tests that the pathogen affects all the crop. Thank you for your attention.